Welcome, everyone. Thank you for being here this evening. This is one of our long-standing partnerships, and I'm very honored to have U of M bring their speakers here 6 times a year. Just a couple things before Chris introduces our speaker. This program is being recorded for accessibility purposes, so at the end of the program, I'm going to carry the microphone around and ask that all questions be spoken into the mic so we can record those for everyone who's watching from home. And we also have hearing assistance available, so if you have trouble hearing the speaker at any time during the program, please just come find me at the back of the event, and we can get you set up with hearing assistance, okay? All right, thank you.
Great, thank you, Jackie. All right, thank you very much, everyone, for coming to the downtown library tonight. My name is Chris Monk. I'm a professor at the University of Michigan here, just down the street in the Department of Psychology. Tonight, I'm very excited to have Dr. Gideon Rothschild, who will be sharing his research on how the brain processes sound. Dr. Rothschild is an assistant professor in the biopsychology area of the Department of Psychology here at the University of Michigan. He's also an adjunct assistant professor at the Kresge Hearing Research Institute and an affiliate of the Michigan Neuroscience Institute. He joined us in 2017 after completing his undergraduate and graduate studies at Hebrew University in Jerusalem, followed by a postdoctoral fellowship at the University of California, San Francisco. Dr. Rothschild explores how the brain makes sense of complex sounds. He uses advanced techniques to measure brain activity in detail, tracking how groups of neurons respond to different types of sounds and how the brain integrates this information. With these tools, he studies how we recognize and understand patterns like multi-frequency tones or sound sequences, and how the brain's sensory and memory systems interact in different states, like wakefulness and sleep. Dr. Rothschild is published in top-tier journals, including Nature Neuroscience, Current Biology, PLOS Biology, and Cell Reports. His research program is supported by multiple active grants from the National Institutes of Health, as well as private foundation grants from the Alzheimer's Association, the Whitehall Foundation, and the Brain and Behavioral Research Foundation. This is a really impressive amount of research support. Please join me in welcoming Dr. Gideon Rothschild, who will be presenting his talk titled, How the Brain Makes Sense of Sound. Welcome, Gideon.
Thanks a lot, Chris. Thanks a lot, everyone, for coming here. I'm very excited to be here and to tell you a bit about the work we do in my lab to try to understand how the brain makes sense of sound. So I would actually like to start with a bit of a disclaimer. Here's a list of the few recent talks in this series of Exploring the Mind. Some really fascinating talks. We've had talks about adolescent mental health crisis, about how fathers matter to the development of young children. Do students really learn critical thinking in college? Really fascinating set of talks. But if you've been coming to these regularly, I wanted to do a bit of a forewarning that my talk is going to be a little bit different, because rather than children, adolescents, or adult humans at least, the study subjects in my lab are rats and mice. And you might be most familiar with rats and mice as something you need to set up traps for so they don't go into your pantry. So I think it's really a relevant and legitimate question to ask, can we study questions of mind and psychology in rodents, which is what we try to do. So I thought I would start with a slide of just examples from the broad field where we, at least as a field, try to do that. And I'll try to convince you that indeed we can, and it offers some trade-offs relative to working with humans. So fear and anxiety, you might have seen those in the titles of the human talks we just saw, can definitely be studied in rodents. In fact, you can teach animals to fear a sound. If you associate a sound with an aversive cue, such as a mild foot shock, they will learn to fear that sound. You can come back the next day or week or month, represent that sound, and the animal will express noticeable fear responses, such as freezing. Rodents are excellent learners and memory formers and have a very strong motivation. They can easily solve various mazes. So rodents will not only solve this maze to get to a food reward very effectively, but they will remember the path that they took the following week and be able to get there quicker. They can, if we put them in these kinds of operant conditioning chambers, they can learn that a specific patterns of light and visual stimuli and auditory stimuli can induce them to press levers so that they can get rewards or avoid punishments, or if you will, rodents playing a video game. Rodents definitely express aggression, and in some cases, aggression that appears somewhat similar to aggression presented by humans. None of us, of course, nobody in this room, but I'll show you a brief clip from a video where the aggression center of this black mouse is being activated by light, and the moment that it is activated will be notified by the title "Lights On." Right, so we see an aggressive expression, aggressive behavior that is not only expressed by these animals, but we can, as in the lab, identify exactly where in the brain that is controlled and how it is mediated. But these mice that might be attacking each other during the day actually express also very strong social behavior. In fact, they like to cuddle up or huddle up before they go to sleep. Incidentally, by research conducted in my wife's lab, Dr. Gideon Rothschild, also in our department. And finally, we have hundreds of strains of rodent animal models that are models of various psychiatric, mental, and neurodevelopmental disorders in humans. Oftentimes, these mice have been genetically engineered to express genes which are prevalent in the human condition. And so they carry the human gene and oftentimes also express issues that are similar to what we see in the human condition. And all of these various behaviors, I would argue that not many people think that they are perfect recapitulations of the human condition, so it's not in a way the real thing, but they do offer a really nice and attractive trade-off. They allow us to study the brain and the neural mechanisms that underlie these psychological and mental disorders in ways that are more invasive and more detailed than we can do in humans. And so that raises the question of how similar our brain is to that of rodents, right? It could in principle be the case that they express similar psychological and behavioral phenomena, but with very different neural mechanisms. And if we compare the brain of humans to that of rodents, at a first glance, it does indeed appear very different. You can see the brain of a human illustrated here. It has these foldings in the outer layer called the cortex, whereas the brain of a rat or a mouse does not have these foldings. The external layer is smooth. And if you compare the sizes, and this is to scale, obviously the rat mouse is much smaller than that of a human, so clear differences. However, if you look in detail in terms of which brain structures are present in each of these brains, we find extreme similarities between the brain of humans and rodents. So humans have the cortex, which you can see here, the outer layer, and so do rodents. Humans have a hippocampus, which we'll talk a bit more about later as involved in memory. So do rodents. Humans have a cerebellum, so do rodents. In fact, you'd be hard-pressed to find a single brain area that we humans have that rodents do not. And therefore, the behavioral similarities and the anatomical similarities, we use those in order to use research in rodents to try and shed new light on the neural mechanisms underlying these psychological phenomena. So how do we go about that? We can study brain mechanisms underlying psychological phenomena at many different levels of resolution. We can look at a whole brain level. For example, if you go to a doctor and you're doing an MRI scan or an fMRI scan, you can look at brain regional level activation. But that is the biggest level of resolution. You can study brain activity and brain mechanisms all the way to the finest small details of protein structure and activity. In my lab, our resolution of interest is here, are networks of interacting neurons that make up the brain. So we humans have about 80 billion individual neurons. These are brain cells that every now and then pulse electrically and send information from one to another. And the aggregate activity of these networks of neurons, which is extremely complex, is what eventually allows us to think, make decisions, remember, act, and I would argue make up who we are. So it's at this level that we conduct in my lab our research, monitoring brain activity of groups of individual neurons and trying to relate them to what the animal is doing, what it is hearing, what it is trying to memorize. And when I say that, I mean that quite literally. So I'll show you a couple videos. A video of a mouse that is going to be on the left. The mouse is head fixed, but it is awake, behaving. It's going to be hearing some sounds that in this case we won't hear. You might even see that it will be getting some milk reward in response to something that it is doing. And on the right side, you will literally see brain activity of groups of individual neurons of this same mouse. So on the right, you're seeing each of one of these circles is an individual neuron, a brain cell, and whenever it lights up, it means it is electrically active at that moment and sending its electrical pulse to some of its neighbors through synapses that we can't see here, but eventually this ensemble network level activity is what we think is the computation that this brain area is doing, and we can do that with current technology in awake behaving animals. All right, so we use the technique that you just saw now. We also use other techniques in the lab, electrophysiological recordings. We won't go into the details of those. We also do neural inactivation. So if we suspect that a brain area is involved in some functions, aggression, memory formation, or hearing, and so on, we can momentarily inactivate that brain area and see the consequences of behavior to determine if this area is indeed causally involved. So what kinds of questions do we ask? At the first level, we're very interested in how the brain represents the external environment. Sensory stimuli like sounds, visual cues, olfactory cues, and so on. And at this level, this is called just sensory processing. But in everyday life, when we hear a sound, or when we hear or we are experiencing a sensory cue, that may guide us to take specific action. Let's say you're at home, you're sitting on your couch, and you hear the music of the ice cream truck from around the block. You might decide to go and get an ice cream. However, this process is further complicated by the fact that it is very context dependent. If you're hearing this music from your TV, same identical tune, you will usually not get up and look for the ice cream truck around the block. You're able to identify the relevant context. And this is further even more complicated because it is very memory dependent. So you may or may not go and look for the ice cream if last week you went and all they had was lemon sorbet, which happens to be my least favorite one, might be your favorite one, but then you may not go, or you may go because it is your favorite one. So there is and obviously whatever you decide to do will in turn also leave a lasting memory trace. So this is a very complex interaction between sensory processing, behavior, memory, and context, which is very difficult and hard to study in the lab, but because it is so prevalent and so reflective of actual natural behavior, this is what we try to do in my lab, try to address and try to study at the neural levels that I discussed. And in terms of the sensory stimuli, the sensory modality that we use the most is sound and hearing, and the studies that I'll talk about today are mostly focusing on the auditory modality. So our menu for today, I will try to give you some just brief views of 3 projects from my lab. A couple of them have already been published, and one is currently in the pipeline. The first one is about how the brain processes sound in motion while we're moving. The second is how the brain processes sound sequences. And the third, the consequences of sound exposure during sleep on memory. So I have to give you just a bit of background about the auditory pathway. So here it is. Sounds arrive at our ear. You can see the cochlea. In the cochlea, we have a very interesting mechanism that translates sound vibrations, air vibrations that arrive at our ear, which are the sounds, and turn them into neural signals. These neural signals then travel up the auditory pathway through various stations, eventually reaching the highest station in the auditory pathway called the auditory cortex. So this is the section of the cortex, the outer layer, that is devoted to sound processing. And as expected from the top of the hierarchy, it is strongly involved in sound perception. How do we know this? For example, if animals or humans that have suffered lesions in their auditory cortex, they do not become deaf. They can still identify that a sound has occurred, but they have issues with identifying complex sound perception. So for example, speech perception is impaired, or hearing in the background of noise. So on the one hand, we know that the auditory cortex is involved in kind of these higher level sound perception processes. On the other hand, exactly how it supports sound-guided behaviors is largely poorly understood, largely understudied. And I would argue that part of the reason is that for many years, this has been the setup where auditory processing has been studied. So animals have been anesthetized and then put in a soundproof chamber where there are no inadvertent sounds that are occurring. So we have very good experimental control in this situation. Sounds are presented and neural activity is measured. And this is a very convenient setup for us as experimenters. The problem is this is not how we hear sounds in everyday life, right? This is not how we guide our behavior based on sound processing. It dissociates hearing from behavior. In fact, in everyday life, we hear while we move, while we act, and in many cases, this is not just beneficial for everyday behavior, but is also, in this case, for example, necessary to survive. Now, one thing that you see in these different examples, dancing to the sound of music, running while listening to your music, a predator or a prey running away from a predator using its hearing, and so on, is the use of hearing while we're moving, while we're walking. And this, until recently, has been completely unstudied due to technical reasons. So in one project, the first one I would like to tell you about, we were asking a pretty simple question: how does the auditory cortex process sounds while we are moving about, or more accurately, while the rodents are moving about? So split it up into two questions. Is the auditory cortex involved in processing sounds while we're moving during locomotion? And if so, how do these groups of neurons that I showed you before process sound in this state? This was a project headed by a former graduate student of mine, now Dr. Carlos Vivaldo, who is currently a postdoc and actually already applying for faculty positions. This was a big part of his thesis project. So we started off by asking, to remind you, is the auditory cortex necessary for sound processing during movement? To do that, we formed a new behavior. Mice were set on this treadmill. Whenever they started running, they would hear a sound, or every now and then a sound would come. And when that sound occurred, it indicated that a reward is about to be delivered. And what we see, we could monitor the individual licks of these animals, and you can see a description of that here. So time 0 here is when sound is delivered, and each tick here is a lick. And there's a gap between the sound and when the reward is delivered of about 1 second. And what you can see here, without going into too much detail, is that immediately after hearing the sound, animals started to lick predictively, anticipating that the reward is about to be delivered, what we call predictive licking. So they were able to process these incoming sounds while running and use them in order to guide their behavior, specifically licking to reward. We then did something very simple. We inactivated the auditory cortex and asked, how does this behavior, how is this influenced by auditory cortical inactivation? So you can see here on the left is control, and on the right is when we inactivate the auditory cortex. Here's a graph of this predictive licking in control. We see this very nice ramping up of licking in preparation to the reward following, guided by the sound. And when the auditory cortex is inactivated, it completely goes away. So the animal's ability to sense the sound while it is running and guide its behavior accordingly has been complete or almost completely eliminated when the auditory cortex is inactive. So this shows to us that the auditory cortex is strongly involved in hearing while moving, something that had not been known previously. That then opened up the question of how do these networks of neurons in the auditory cortex process sound while moving? To do that, we used a very similar experimental setup as I showed you before. The animal can run or not while we measure neural activity of groups of neurons in its auditory cortex. So I'm just going to describe what we're seeing here, and because we're going to see a few of these coming up. So this is a response of one neuron to the same sound, multiple repetitions. Sound is denoted here in gray, when the animal is running in green, and when the animal is immobile in red. Y-axis just denotes how active it became following the sound. So you can see that this specific neuron responded or represented the sound that it heard in both immobility and in locomotion. Here are a few additional examples, and we found that some neurons respond stronger when the animal was stationary, and some responded more when the animal was running. So we found these subgroups of neurons that seem to be dedicated to hearing while running, and others that seem to be dedicated while immobile. But really the most surprising finding that we found is as follows, is not what we were looking for. Here's what we saw. So what you can see here are overlaid two signals. One is the speed of the animal in green, how fast it was running moment to moment, and in black is how active a specific neuron was. And you can see there's almost a perfect overlap between the two. So in other words, and there are no sounds presented here at all. So what we found was that in the auditory cortex, a brain area that we previously thought is mostly dedicated to sound processing, what we actually see is very strong representation of the animal's own running speed, something unrelated to sounds altogether. We used statistical models to show that we can decode the speed of the animal from the activity of these neurons. So I'll skip this slide. Put together, what we found in this study is that rather than just representing sounds, by recording activity in animals that were running, we found that the auditory cortex integrates sound with the animal's own running. And we believe that this might be very important because, for example, in some of these behaviors, such as dancing to the sound of music, the brain must integrate the music that is coming in, the sounds, and the person's own movement in order to guide its future behavior. So the auditory cortex is important for integrating sound and movement. I'd like to tell you now about another study that we did in the lab. And to do that, I would like to introduce you to, I didn't say much about the kinds of sounds that we've been presenting. I just told you that we were presenting sounds in the first chapter. The most common sound used in auditory neuroscience are these kinds of sounds. They're called pure tones. Oops. Okay, so they're very synthetic, very unpleasant, sorry about that. They're very common, but yet also very unnatural in everyday life. You basically never hear a sound of this sort, and for various reasons, it has been the most common stimulus used for auditory neuroscience. And when you present these kinds of sounds, what you typically see is that as you present the sound, that's this gray bar here, a neuron might respond with increasing its electrical activity. And you can see that it does that immediately after the beginning of the sound, after the tone comes on. So the tone comes on and the neuron responds very robustly. Okay, but real-life sounds are rarely like this. They often come in sequences. Here's one. That is a warbling vireo. Here's another one. Anybody know what the animal is? A guinea pig. Yes, nice. How about this one? Does anybody identify this sound sequence?
I'm going to make him an offer he can't refuse.
Anybody from where that is? Godfather, great. Right, this is Marlon Brando in The Godfather. I'll just play it again because I love that.
I'm going to make him an offer he can't refuse.
I'm going to make him an offer he can't refuse. And the final sequence. All right, so sounds in everyday life often don't come isolated. They come as sequences. And the meaning of the sequence often depends on its entirety, right? And that's a point I would like to further make using this example. So here is the sentence that we saw in the previous slide. I'm going to make him an offer he can't refuse. So I told you that we're looking in the auditory cortex as representing sound meaning. So at what moment in time, if we were to record neural activity, should we look for the meaning of this sentence? If we were to treat it like an instantaneous stimulus, so then we should do it just at the beginning. But clearly, the response to the word I'm doesn't tell us what the meaning of the sentence is going to be, right? It's going to be, I'm going to make him a cocktail or something like that, right? When is the first moment in time that we have the complete meaning laid out of this sentence? Only in the end, right? Only at the end of the sound sequence do we know with certainty what the meaning of the sentence was. And those are times where there's not been much research about responses after sounds, but mostly during sounds. But because of the reasons I've mentioned, we hypothesized that this moment in time after a sound sequence has terminated, that's a great candidate time point in which the brain can decode what has just been heard and what is the meaning of it. That's what we set out to do. And to our hypothesis was that neural responses to the termination of sound sequences, something we call off responses, represent their acoustic history and behavioral salience. So we set that in the spirit, so this was research done by my former postdoc in my lab, Dr. Jun-Yup Lee. And in the spirit of my introduction, we did this in rodents. So we used very simple sequences to start with, although now we're using some more complex ones. So they're just sequences of either AA, AB, BA, or BB, where each letter is just a tone. Okay, so just pairs of sounds. So the first thing we asked was, do we indeed see these responses to the termination of sound sequences, and do they tell us retrospectively, what the sound sequence was? So here's an example of one neuron. Its responses to the sound sequence AB in blue, and its response to the sound sequence BB in red. And hopefully you can see that after the sound sequence has terminated, this is after the gray bar, we see a differential response, responding bigger to the BB sound sequence than to the AB sound sequence. Even though at the moment of this response, there is no more sound any longer. So this tells us that this specific neuron could tell us retroactively in time, did I just hear AB or BB? Here's another example of another neuron differentially representing the sound sequence by these off responses, responses to the termination of sound sequences. And we use statistical models to show that you can indeed decode the identity of the sound sequence from the responses to their termination. Okay, great. So off responses encode sound sequence identity. What about its meaning? To address that, we now taught animals that one specific sound sequence means reward is coming. So without going into too many details, we just presented a sound sequence, AB, followed by a second of silence, and then reward was delivered. We did that across many presentations, at the end of which the animal knows that AB means reward is coming. So we have now given this AB sequence a positive appetitive meaning. And we were interested in asking, how does this now change the responses to this AB sound sequence from before to after it has gained meaning? So here is an example, or two examples of individual neurons, how they responded before the sound sequence gained meaning versus after. And you can see that in both of these cases, the response to the off response to the sound sequence has dramatically increased. And you can see that even in these two as well. So what we see indeed is that these responses to the termination of sound sequences don't just tell us what was just heard, but also the value of it in a way, the valence associated with it. All right, so auditory cortex responses to termination of sound sequences represent the sequence and its salience. All right, so the last project I would like to tell you about, this one is still unpublished, although hopefully it will be soon, is a bit different. It's about the consequences of sound exposure during sleep on memory. So I've showed you a couple of examples and kind of try to illustrate with a few others that processing sounds in everyday life is really important to guide adaptive behavior, right? So we can use sounds to decide what to do, in a way that benefits us. What is much less studied is that the brain also hears while we're asleep. So until 25 years ago, it was thought that while we're asleep, the brain is essentially deactivated, and that the role of the thalamus, brain area in the middle of the brain, is to block sounds from the responses to sounds from reaching higher levels. That has turned out to not be true. In fact, the whole auditory pathway is never asleep, so to speak. It responds very robustly even when we are asleep. And of course, we all know that you can wake up from an alarm clock, so in that sense, it is not surprising. But classical studies have shown that not only does the brain respond to external sounds, but also to their meaning. So studies have shown that if a person is being whispered the sound of their own name, they will tend to wake up more often than if a different name is whispered to that person at the same level of intensity. So these studies and others have in response to them, people have called the auditory system the sentinel that stands guard while we are asleep. So it is constantly active, monitoring the environment, even when we are not conscious. But I think that because we are not conscious to all of these sounds when we wake up the next day, it has been not so motivating to study what happens when we are exposed to sounds while we're asleep. So this is something we were really interested in this project, and we acknowledged very early that this ability to process sounds and their meaning while we're asleep is very evolutionarily beneficial, right? It's really good to us that we can hear the sounds of our baby while we're asleep, so we can wake up and tend to them. And in fact, I remember that when I first became a dad, the sensitivity to these external baby cry sounds increased very much so. Now less so as they have aged, but clearly this is very beneficial and evolutionarily adaptive. It's also really important for us to be able to hear a source of danger while we're asleep so that we can wake up and take precaution. So in that sense, it makes a lot of sense that our auditory system is constantly monitoring incoming sounds. The question we were interested in this project was, does that come at a cost? So is there a cost to the fact that our auditory system is constantly involved in gauging what is happening from an acoustic perspective? And it is motivated by the fact that most of us do not live in the savannah and are kind of at the danger of lions or tigers, but are more likely to live in an environment like this. So this is not exactly Ann Arbor, but let's say we live in Manhattan or in other noisy environment. If our auditory pathway is constantly monitoring the acoustic environment, does that come at a cost to what the brain should otherwise be doing while we're asleep? So what should the brain otherwise be doing while we're asleep? A key function of sleep, as we see it, is the process of memory consolidation. That is the process of taking the day's events and making long-term memories out of them. That is something that we know strongly depends on sleep and very strongly relies on the activity of a brain area called the hippocampus. The hippocampus, while we're asleep, is reprocessing the day's events and crystallizes them across time to become long-term memories. And we were interested here in the question of when sounds are coming in while we're asleep, does that interrupt this process of the hippocampus to consolidate the day's events into long-term memories? So here's the hippocampus in a 3D animation in a human. It's this red structure in the center. Just a word of background about the hippocampus, if you've not heard about it before. Probably the most famous and a classic study about the hippocampus come from the patient Henry Molaison, which up until he passed away in 2008 was known in the literature as patient HM. Henry Molaison suffered from epilepsy as a child, probably as a result of a bike accident, and this is in the 1950s. There were not as sophisticated as approaches to deal with epilepsy at the time. Doctors identified that the epileptic center in his brain resided in the hippocampus. And with the tools that were available at the time, what the doctors did was completely remove the hippocampi from both of his hemispheres. And in terms of the epilepsy, that kind of worked. So his epileptic seizures were strongly reduced. But as a result, patient HM lost the ability to form new memories completely. So he was not, you could have an interaction with him, there's a conversation, come back the next day, he would have zero recollection that you had ever met. And dozens or hundreds of studies since in humans and animal models have recapitulated this, really highlighting that the hippocampus, this red structure at the center of the brain, is absolutely critical for the brain's ability to take the day's events and make out of them long-term memories. All right, so we were then interested in asking, does exposure to sounds while we're asleep, sounds that don't wake us up but clearly still take up neural processing power, does that interfere with the hippocampus's role in memory consolidation? This project was is still headed by my postdoc, Dr. Carla Salgado Puga, and as I said, hopefully will be published soon. So Carla recorded from the brain, actually the hippocampus of rats while they were asleep and experienced various sounds coming in. So what we noticed very quickly is that indeed presenting sounds to sleeping rats interfered with the activity of the hippocampus. So you can see an illustration of a rat here. Its activity from the hippocampus is being recorded. It's a naturally sleeping animal. And I won't go into all of the details of what you see here, just to say that on the left, you see a recording from the hippocampus in the first hour of sleep when no sounds are presented. And depicted here is a summary of neural activity in the hippocampus. And this red blob here denotes a really important neural activity pattern in the hippocampus. After an hour of unobstructed sleep, sounds were presented, and as a result, this red blob completely went away. So neural activity in the hippocampus was really strongly suppressed, which made us then ask, okay, if the hippocampus, which we know is really important for memory consolidation, if its activity is really being suppressed by sounds, how does that influence memory formation, the function that the hippocampus supports? So to do that, we devised a memory task in rats and asked how well or how does sound presentation during sleep influence the animal's ability to remember what it learned. A couple of details about the task. So the rat is put in a 3-chamber setup. It can choose to go to the right side or the left side. And wherever it chooses to naturally go to, we put sugar water on the other side. So we try to teach it that the side that it originally preferred, it should switch preference to get sucrose on the other side. We do this across multiple presentations. As I told you in the introduction, rats are very fast learners, especially when food is involved. So they will quickly, in baseline conditions, shift their preference to the side where they know that sugar is delivered. The animal is then going to sleep for 3 hours, and this is where the animal will either sleep unobstructed or we will be presenting sounds to test their influence on memory consolidation. And then we retest the animal's memory. And the way we do that is we just put the animal in this 3-chamber setup and look where it goes. If it goes to where sucrose was previously presented, we infer that the animal has remembered, has retained that memory from the learning. So I won't go into everything you see here, but basically the y-axis, the top part, is a measure of memory retention. So if you look after 3 hours, immediately after the animal woke up, you can see strong memory. This depicts the tendency of the animal to go to the side of the chamber which we have associated with sucrose. So almost all animals have shifted their preference to this side, indicating they remember where sucrose is delivered. If you look 24 hours later, they still remember which side they got the sugar in. And this is in baseline conditions. No sounds were presented in sleep. Now we want to compare this to what happens if during this intermittent sleep of 3 hours, we presented the sounds that we know suppress hippocampal activity. Then we see what we see on the right. So after 3 hours of learning, the animals seem to remember which side they got the sucrose in, but 24 hours later, they have completely forgotten. So you can compare that to the control on the left. So they have completely reverted to their original preference and completely forgotten what they had learned. So what this tells us is that presenting sound in this sleep session after learning has essentially completely obliterated or completely impaired the animal's ability to remember that event, that learning process. So our conclusion from this, and with a few grains of salt, is that sound presentation during sleep suppresses hippocampal activity and memory consolidation. And it is really important for me to put these asterisks here. It is not all sounds that do this, and we can, if you're interested, we can discuss which sounds might actually do the opposite. It is not all memories that this might do, in which we might see this effect, and this is rats. So I've often gotten questions about my children or my parents. This is all only with respect to rats in our labs so far, although I do think that these phenomena should motivate us to look at the human condition as well, especially because we know that exposure to noise in urban environments is on the rise and has been shown to be associated with development of various disorders, including early dementia and higher risk for Alzheimer's disorders. So whether there is something similar in humans still remains to be seen, but our study shows that, at least in rodents, it does seem to be a really serious risk factor, and I would recommend to maybe try and avoid it just in case. All right, with that, I would like to thank the people who did the work, the members of my lab. I try to highlight the people who were involved in the specific projects as we went along, and also to the funding agencies that made this work possible, and thank you very much for your attention.
Thanks, everyone. I'll take questions now. You got one?
The experimenting with mice or rats during sleep
Yep.
was very interesting. My question is, if we can infer it to humans, the definition of sleep is kind of complex, because when you have the altered state of consciousness, just when you start to get into sleep, the functioning of the hippocampus might be different than when you're in deep sleep. So my question to you is, what do you think would happen, for example, that if you were laying on a couch and you're watching TV and you're starting to get into a light stage of sleep, and something on television, which so many people listen to, is something that violates your morals or your principles or something, would the hippocampus function differently in its memory aspect than if you were in a deeper stage of sleep?
Yeah.
I know we're kind of stretching this out to humans, but that makes it interesting.
Yeah, no, it's a great question, so sleep is, exactly as you said, has multiple stages. The very first distinction is between rapid eye movement sleep, that's the stage where we dream in, and non-rapid eye movement sleep. I didn't mention it here for time purposes, but the sounds that we presented were presented selectively during non-rapid eye movement sleep, so not while we are dreaming, and also only after the animal has reached a pretty deep level of sleep. So the effects that we see here are, at least so far what we've done, only reflects presentation of sounds that don't wake the animal up and are presented while we're in deep sleep, in the stage where we're not dreaming. Now, if we hear it in lighter sleep, what might happen is that we have a higher chance of waking up. And if we wake up, that kind of, it makes the result a bit more complex, because we know that interrupting with sleep impairs memory. But that is kind of for a potentially different reason. So in this study, we were particularly interested in presenting sounds in the deep sleep, because those are sounds that don't wake us up. And then when we when we've woken up in the morning, we have no recollection that anything happened. And what we see is that even those sounds impair memory. So if you're, I don't know, if you're exposed to sounds that are either so loud or you're hearing them when your sleep is pretty shallow and you're waking up, then of course you're going to have memory impairments, but that's probably because your sleep has been interrupted.
I guess my question was the sounds that you hear during the early stage to humans, what if the sounds that you heard were negative sounds versus positive sounds?
Oh, I see. Okay, that's a great additional question.
Well, I mean,
So in this, another thing I did mention is that in this study, the sounds were presented were neutral sounds. They didn't have any associated meaning with it. We are 100% convinced that the valence associated with the sound will have an influence on the memory impairments resulting from it, and also the tendency of the animal to wake up. So much so that we asked the NIH to fund a 5-year project looking into that question, and we were lucky to be funded. So hopefully in 4 years I can come back to you with some answers.
So considering mental health and the risk of dementia, Alzheimer's,
Yeah.
For people who live in the city and have that inevitable sound, are earplugs helpful, or does it still impact the body despite earplugs?
Yeah, so again, I have to warn, any generalization to humans and so on is a big maybe.
Your best guess.
Yeah, my best guess. So my best guess is as follows, and is that first of all, if you live very regularly in an environment where the sounds are very regular, your brain would, this is my guess, develop some level of adaptation and resilience from this. So in this study, the sounds that we presented were novel to the animals and unpredictable. If you were listening to the sounds of your dishwasher, which is very repetitive, very regular, it might not grab the brain's attention as much while we're asleep and may have actually opposite consequences. In fact, there are now machines that present constant white noise that is a sound that you hear, but it's completely predictable, and what its effect is oftentimes very beneficial, at least for sleep, because it masks other sounds. So if you put earplugs, you'll be eliminating any sounds, but it might make you more kind of susceptible to surprising loud sounds that go through. Whereas if there's a baseline hum, that might not happen. So yeah, I don't know, but I would speculate that if you're living in an environment, if you're only there for a short amount of time and sounds are very intrusive for you, wear earplugs. Otherwise, it might not be beneficial. Yeah, good question. Other questions?
I heard that sometimes patients with Alzheimer's disease do something, memory tricks when they hear music. Why is that?
Okay, so you mean that, sorry, just to make sure I understood the question, that you've heard of a treatment to treat Alzheimer's disease patients with music?
Yes, sometimes they kind of respond to the music from the 20s, and they kind of, like react more briskier. So why is that?
Right. Okay, so it's a great question. A lot of this is unknown. So musical therapy is used as attempts to treat multiple disorders, not just Alzheimer's disease, also Parkinson's disease. And with Parkinson's disease, for example, it's actually showing very positive effects in many patients. Exactly why it works, we still don't know. We're investigating. In Alzheimer's disease patients, one thing that is very interesting, and this is also relevant to patient HM and other patients who've suffered disorders in their hippocampus, is that musical abilities, musical knowledge is not dependent on the hippocampus. So in fact, there's another famous patient called Clive Waring who has also suffered a complete lesion of their hippocampus and remained a super talented piano player, and his ability to play very complex pieces was not impaired, even though he suffered not Alzheimer's disease, but an extremely severe case of memory loss. So I'm not 100% sure the specific case you're describing, but it might be that in Alzheimer's disease patients where episodic memory is impaired, their ability to remember recent events, where that is really impaired, triggering memories using musical pieces which are not dependent on the same structures might be beneficial. That would be my best guess.
Any more questions? Oh, okay. Thank you so much for a wonderful presentation.
Thanks.
Thanks so much.
Yeah, thank you.
That's really great.
Thanks.
So hearing about HM, it's what really got me